Hi, my name is Mark Anderson. I'm the William Osler Professor of Medicine and Director of the Department of Medicine at Johns Hopkins University. And it's my pleasure to tell you about what we think is a really interesting story that will be published in the Journal of Clinical Investigation. This story was led and directed by a very talented postdoctoral fellow, Kletos Konstantinidis, who is a physician scientist and completing his second year of clinical cardiac electrophysiology training. It's also a wonderful collaboration of many people, but I think I would like to just point out here Rod Levine, who is an expert in methionine oxidation, whose laboratory is at the NIH, and Alex Kolodkin, who is an investigator in the Department of Neuroscience at Johns Hopkins University and co-discovered an enzyme we'll talk about called Mical, a methionine oxidase. So I show here a cartoon of CaMKII. CaMKII is a serine threonine kinase that pictured as a monomer. The holoenzyme exists as a dodecamer, where there are two pairs of 6 enzymes joined together by the green C-terminus association domain. Now it turns out that CaMKII is mostly inactive in cells. It's richly adorned in cardiac cells, neuronal cells, immune cells, excitable cells. And its initial activation occurs when calcium rises in the cell and complexes with a ubiquitous calcium-sensing protein called calmodulin. Calmodulin here is depicted as that little orange dumbbell. And when calmodulin is calcified, it changes its conformation and embraces CaMKII in a highly defined calmodulin binding region in the C-terminus of this purple part, the mid part of CaMKII that's shown here, the regulatory domain. And once that happens, it creates a conformational torsional change, allosteric change that prevents the regulatory domain from constraining or auto-inhibiting the catalytic domain, shown as this orange-pink part. And it pops open, if you will, and then the enzyme becomes active. Now, it turns out that the enzyme can be further modified by post-translational events that are pictured in the far right panel as modifications, that little orange lollipop on the N-terminus region of that purple regulatory domain. They all kind of work in the same way. They change the charge structure of the autoinhibitory region and prevent CaMKII from being able to auto-inhibit once calcium has gone down to a basal level in the cell and calmodulin binding is reversed. A number of years ago, our group, in an effort led by Jeff Erickson, who's now at Otago University in New Zealand, discovered that a pair of methionine residues in the regulatory domain could be oxidized on CaMKII. And when they were oxidized, that trapped CaMKII, like other post-translational modifications, into a highly active and conformation. And it's now known that oxidized CaMKII contributes to a number of major diseases that appear where excessive oxidant stress drives the disease. Diseases like heart failure, arrhythmias, asthma, vascular disease, and cancer, to name some. So we're really interested in what could make this work. And so there are a couple questions left open in the field. One was, given the holoenzyme nature of CaMKII, why didn't physiological levels of oxidant stress lead to a feed-forward situation where CaMKII was hyperactivated and always led to disease? And another question related to the slow time constant of methionine oxidation. So, for example, in contrast to cysteines, which are promiscuously and rapidly oxidized, methionine oxidation is slow. And so one question had been, does it need to be directed catalytically by an enzyme? And that's kind of where our story starts. It's worth noting that this methionine pair that we discovered had functional properties is a pretty recent invention in vertebrate evolution. So CaMKII as an enzyme is ancient. It's about 1 billion years old. And this twin pair of methionines, which are pictured as the yellow stripe, they first arose in the primordial vertebrates, and then they were sustained throughout subsequent speciation. And we're working to to understand why CaM kinase took on this burden that created the potential for adult diseases. And so when we worked with Rod Levine at the NIH, we first were expecting that we would find an enzyme that modified this pair of methionines. And just as a spoiler alert, I'll tell you, we still don't know what does that. But I wanted to focus in this thin red box, or here in this expanded version, to say that we ended up focusing on another methionine entirely. And this is one in position 308 that lies right smack in the middle of the calmodulin binding domain. And so this one is different in that not only its position and the functional domain where it resides, but also because it is ancient. It is part of the primordial CaM kinase. CaM kinase never existed without it, and it exists in all known isoforms and species of CaM kinase. So we first used a candidate approach, reasoning that methionine oxidation, if it were to be directed by enzymes, we would first start to look for enzymes that were known to do this task. And the only family so far known to do this are called Micals, or molecules interacting with Cas-L. And what you can see is that there's 3 of these isoforms. The best studied one, the best understood one is MICAL1, and so that was our initial focus. And to say that although these enzymes were discovered 20 years ago, their only known target is actin, where they catalyze the oxidation of a pair of nearby methionines, and that oxidation status sets the balance between filamentous and globular actin, allowing cell cytoskeleton to be dynamic. So we used a variety of techniques, from proteomics to molecular biology to making multiple animal models using mice, using Drosophila, and using human-induced pluripotent stem cells to probe the role of this methionine oxidation. In the end, we discovered that MICAL1 closely and exclusively targeted methionine 308, and that when methionine 308 was oxidized, that it was much more reluctant to bind calmodulin, and so CaM kinase wasn't activated. In the end, this or activated as readily. In the end, this explained why oxidant stress, even at low levels, was unable to create excessive CaM kinase activity, because oxidant stress played a dual role, both to check CaM kinase activation and to activate it at the paired methionines. We also mapped a pathway where a reductase, a stereospecific reductase called MSRB, methionine sulfoxide reductase B, was the partner of MICAL1 to reduce the oxidized methionine 308. We found that if you replace methionine with any residue, including valine, that it acted like oxidized methionine, and it wasn't as good at binding calmodulin. And so we made knock-in animals after validating the constants of binding in naked proteins, and found that Drosophila, which have only one CaM kinase gene, and where if you knock it out it's lethal, they behaved in a hypofunctional way, where their heart tubes didn't contract as well, and their circadian rhythm, a kind of biology under partial direction of CaM kinase, was distorted. For mice, we found that core physiologic functions like heart rate was slowed in mice that had their methionine 308 replaced with valine. And we also examined the MICAL1 protein carefully, and were able to use structural modeling under the direction of Mario Bianchet at Johns Hopkins Department of Biophysics, where he found a mutation that made Mical only target CaM kinase and not actin. And we made mice that had this knock-in mutation, and those mice were not hypersusceptible to injury. In contrast, mice lacking MICAL1 as knockouts had excessive chem kinase activation at baseline and did particularly poorly under pathological stress like transaortic banding. So this is a story of an unanticipated finding that helps to explain the physiologic and stable response of chem kinase to oxidant stress, and we think opens up a new biology to try and understand how enzymes with key methionines may be regulated upstream through a coordinated, orchestrated approach system involving micals and perhaps other enzymes, rather than being the promiscuous victims of oxidant signaling within the cell. Thanks for your attention, and I hope you read our paper.